My name is Ghassan Abu Alfa. I'm from Memorial Sloan Kettering in New York. And I was tasked, actually I would consider it a very great honor with the session on the Research Fellowship Report. First I start by thanking the Foundation and congratulating the Foundation on really incredible work over the many years. And specifically, in regard to the fellowship that, as we know, has been awarded for the last 4 years, and the total awards to date are close to $750,000. And we had, from 2015 till now, a total of 15 awards, and that were funded by or to 12 institutions. I really like to especially name the awards because they were really very important, and we want to really be very grateful and thankful to the families who provided the support for the program in memory of their loved ones. And I specifically mention here Andrea Marie Fouquet Memorial Research Fellowship, Andrea Scott Memorial, Elizabeth Dardan Stefanini Memorial, Jacques DuPont Memorial, Melissa Manza Memorial, Christopher J. Wilkie, and Tommy J. West Memorial. And this year, we are very honored in this report of updates to welcome 4 speakers. And I would like to welcome all of them maybe to the podium, or maybe there's no place for all 4 of you. But tell you what, why don't please, Dr. Aoki, you can come first and then we'll do that. So let me introduce Dr. Aoki. So Sushi Aoki is postdoctoral fellow at the Steele Laboratories for Tumor Biology at MGH, at Mass General and Harvard. And before going to Boston, he worked in the hepatobiliary, as a hepatobiliary surgeon for 10 years in Japan. So he really carries quite a bit of expertise. And he actually is the winner of the 2018 Tommy J. West Memorial Fellowship. And he was going to talk to us about the role of PIGF and RP pathways in intrahepatic cholangiocarcinoma. Please. Thank you.
Yeah, thank you for kind introduction. It's a great honor to have an opportunity to present here. So today, I would like to present my ongoing research, which is blockade PLGF inhibit ICC progression by targeting tumor and stroma interaction. So. Yeah. So the specific character of intrahepatic cholangiocarcinoma, ICC, is abundant of desmoplasia, which is almost same as pancreas cancer. So my hypothesis is that ICC progression and treatment resistance is mediated by the desmoplastic stroma. So recently, several genetic alteration has been reported. So nearly 40% of obtain genetic alteration for potential therapy target. So several molecular target therapy, which is FGFR2 targeting, or IDH targeting, or BAP1 targeting, have been developed in clinical trial. About VGFR2 targeting therapy, this therapy has been shown no effect to pancreas cancer. So we are now focusing on another VGFR factor, which is placental growth factor. We called it PLGF, a neuropilin pathway. In our laboratory, PLGF neuropilin pathway inhibit growth and spread of medulloblastoma several years ago, which work lead now in phase 2 clinical trial in medulloblastoma patient. So now, today the aim is to evaluate whether inhibition of PLGF neuropilin-1 pathway suppress ICC progression. So this is my talk, this is outline of my talk. So firstly, we check PLGF and neuropilin-1 expression in ICC. Neuropilin-1 is a specific receptor of PLGF. So in human rejected ICC sample, we can see over 90% of patients has highly expression of PLGF and neuropilin-1. And in cell line, PLGF is produced by both cancer cell, ICC cell, and also HSC stroma cell, which is hepatic stellate cell. And also in neuropilin-1 expression, we can see highly expression of neuropilin-1 expression in both mouse cell line and human cell line. So as a result, PLGF and neuropilin-1 are highly expressed in human and mouse ICC. So we usually use orthotopic mouse model of ICC. We inject mouse cell line into the liver and follow the liver tumor sites by ultrasound. In material, we use two kinds of ICC cell line. One is a 4-5 with P53 plus KRAS mutation, and the other is SS49 with IDH2 plus KRAS mutation. Using these orthotopic mouse model, we check tumor growth curve and overall survival. After pharmacological PLGF blockade, which is the use of 5D11, which is anti-PLGF antibody, and also genetic PLGF knockdown using SH cell line. So both of them, which mean orange and purple line, both of them show significantly delayed of tumor growth curve and prolonged overall survival compared with control group. Control group means black line. And very interestingly, red line, which means additional pharmacological anti-PLGF treatment with genetic PLGF knockdown. Yes, further show significantly delayed tumor growth curve and prove overall survival. So which means stroma-derived PLGF is much more critical for ICC progression in liver. And in liver lymph node metastasis and lung metastasis, PLGF knockdown significantly reduce of these metastases. So PLGF blockade inhibit tumor growth in orthotopic mouse model of ICC. So next, using these mouse liver sample, we check Ki67 positive cell, which means which reflects cell proliferation. So PLGF blockade significantly reduce Ki67 positive cell, which means PLGF blockade reduce ICC cell proliferation. And also in vitro, PLGF blockade downregulate AKT signal. AKT signal is commonly a driver of cancer cell proliferation. Regarding stroma component, we check SMA positive cell, which means cancer-associated fibroblast, CAF. So CAF reduced by PLGF blockade, like this picture. And we isolated CAF cell line from mouse liver tumor and confirmed highly expression of alpha SMA. And we confirmed that this CAF cell line has very enrichment of NF-kappa B signal-related gene by RNA sequence data. So we add PLGF recombinant and recombinant PLGF activation, NF-kappa B signal, and AKT signal, and also SMA expression. And on the other hand, 5D11, which is a blockade of PLGF, reduce NF-kappa B signal and decrease of alpha SMA expression. And also 5D11 decrease fibroblast-associated gene after treatment. So these desmoplastic disease was associated with opening the vessel by evaluation of IHC, the upper picture, and lower picture shows the OFDI picture of functional blood vessel. This OFDI picture shows PLGF treatment showed increase of blood perfusion into tumor. And as a result, PLGF blockade improves hypoxic microenvironment. So these data shows PLGF promote ICC cell proliferation through AKT activation, and PLGF blockade inactivate CAF through NF-kappa B signal. And PLGF blockade reduce desmoplasia and restores blood perfusion. So finally, we check combination therapy of PLGF blockade with standard chemotherapy, which is gemcitabine plus cisplatin. Combination therapy of PLGF antibody plus gemcitabine plus cisplatin significantly delay tumor growth and prolong survival compared with anti-PLGF alone or gemcitabine plus cisplatin alone. So in short, PLGF blockade enhance ICC sensitivity to standard gemcitabine plus cisplatin. So this is my research conclusion. PLGF neuropilin-1 inhibition delays ICC tumor progression and reduce tumor desmoplasia and hypoxia. So this schema shows PLGF is produced by both ICC cancer cell itself and also stroma cell, which is CAF. And PLGF activation both cell through AKT or NF-kappa B signal. So inhibition of PLGF reduce desmoplasia and increase blood perfusion into the tumor, and finally enhance chemosensitivity. So we are now focusing on the combination therapy with immunotherapy, which is anti-PD-1 or anti-CTLA-4 therapy, because this effect improves immunosuppression microenvironment. Yeah, this is the last slide. I'm really appreciate all support from Koranjo Foundation, and also my friend in my lab, and also my collaborator around MGH Cancer Center. Thank you very much.
Great. Excellent, thank you very much. So we'll carry on to the next presentation. And it's a great honor to welcome Dr. Iris Chehui Luke, who is a postdoctoral fellow at Dr. Supriya Saha's laboratory at the Hutch in Washington. Iris received her PhD in interdisciplinary oncology in British Columbia, at the University of British Columbia in Canada. And her work has focused on mutant IDH in intrahepatic cholangiocarcinoma, and explore novel synthetic lethal therapeutics that can effectively suppress these tumors. And again, I note that Iris was the recipient of the 2018 Andrea Mary Fouquet Memorial Research Fellowship. Please.
Thanks for the kind introduction, and it's really my honor to be here to present my work and also to receive the award. Really thanks to the foundation and all the donors for your support that make this work possible. And so today I'm gonna talk to you about my project, which we are developing novel human-derived model system in ICC. And this is an early stage project that I'm gonna show you about some new data, early data, and also followed by some background. So before I start my talk, I want to give you guys an update about my mentor, Dr. Supriya Saha. He unfortunately cannot come to the meeting because he was diagnosed with myeloid fibrosis, which is a type of blood cancer that associates with bone marrow failure. And he is now undergoing bone marrow transplant in Fred Hutch, which is actually one of the best centers to do this procedure. Although it's a very challenging path for him, but he just wants to update everyone that he is staying strong and he is recovering well. So let's hope for the best for him. So in the SAHA lab, we are focusing on primarily IDH mutant ICC, which is the most common mutation in the ICC context compared to other existing known mutation. And this is also a therapeutic actionable subset of patients. And in our lab, we focusing on multiple area relating to this subset of patient or tumors, including identifying the downstream target of mutant IDH, which may help us to understand how the clinical compound works in the clinic. Second, we are studying the mechanism of the dasatinib hypersensitivity in this subset of ICC. We are also exploring the immune surveillance in mutant IDH using genetic modified mouse model. Oh, sorry. And finally, we are exploring patient-derived tumor slice culture system that it can be used as a rapid drug testing system for this subset of ICC, and also can be utilized as a tool to validate the mechanistic finding of all the three aspects of our projects. And so IDH, many of you know that it is a key metabolic enzyme, and mutation in IDH result in the gain of enzymatic activity of this molecule, result in production of 2-hydroxyglutarate, 2-HG, which is a well-known oncometabolite, and it was shown by Shoup previous work that it inhibits hepatocyte differentiation. And with additional oncogenic hits, it can induce the formation of ICC. However, how IDH inhibitor works in established ICC is not fully defined. And here is the recent phase 2 clinical trial of AG120, the mutant IDH1 inhibitor, and this slide was shown by Dr. Goyo's talk. We can see that about half of the patient result in stable disease after treating with this drug. And this work was presented in 2017 by AGIOS in an AACR conference. And this study was able to obtain a small subset of patients that have pre-treatment and on-treatment biopsy. And they found that patients that performs better in better progression-free survival, they are associated with increased cholangiolar morphology, as shown in the bottom right image. And they are also associated with higher hepatocyte gene expression. However, how this drug exactly work in the tumor is poorly defined, and this is mainly based on the fact that there is a lack of a representative model in this tumor type. And so in the SAHA lab, we have explored a number of different model systems, including in vitro cell line system, in human and also mouse models, and also in vivo mouse system, including genetic modified mouse model, and also a syngenic grafting model that we can look into the pathogenesis. And finally, we are currently developing a patient-derived models, including tumor slice culture model that we're going to talk about, and also PDX model as well as organoid culture models that can be used as a therapeutic to measure the therapeutic response. And so in the tumor slice culture system that we started to develop is in collaboration with Dr. Raymond Young in University of Washington, which is the first cancer surgeon that provide us the ICC resected tumor. We were able to obtain the tumors and then cut the tumor core into very thin slices and then plate them into culture plates with some inserts with pore in the bottom. So these slices can grow for a period of time, and then that allows us to treat them with multiple drugs or in combination of drugs. And then we can monitor the change of or the response of the slices to various drugs by a number of readouts, including MTS assay, which we measure the change in the slice viability. And also we can get the histology and take the slices for immunohistochemistry, looking at the proliferation marker and also cell death marker. And we can also get the slice for cryopreserve for RNA and protein further analysis. And we also be able to genotype the IDH mutation status of patients. And here is an example of the how the slice culture looks like in after like 3 days of culture. On the left is the H&E staining that you can see is a very typical ICC morphology with a lot of stromal component. And on the right is the Ki67 proliferation marker staining in brown. You see the cells are proliferating. And we were able to show that the slice in culture can maintain viability on up to 28 days by using a special media on the left graph in the right line. And on the right, you can see the morphology at day 7 and day 28 of the slice after culturing, and they actually still retain quite a lot of original tumors morphology. And so this is one of the example of how we measure the slice culture response to multiple drug treatment. And this is one case of IDH wild-type patient that we cut into multiple slices, and then we subject them into various drug candidate treatments. And we first, at day 0, we measure the pre-treatment viability, and then we treat the drug for about 3 to 4 days, and then we measure again the viability to look at the change. And the graph on the left, you can see is a change of viability. On the left is the DMSO vehicle control, and on the right is STS, which is cell death control, that you can see 100% of reduction in viability. So the drug we test, you can see here, they are somewhat between the positive and the vehicle control. We so far obtained about 14 cases of tumors tissue, and we genotype them, and all of them are wild-type, IDH wild-type tumor. And we are still waiting for the first IDH mutant tumor. And here are more examples of how the tumors look like in H&E and also IHC staining. On the top are the DMSO treated, and on the bottom are the positive control, STS treated, which is apoptosis inducer. You can see that in the DMSO, the Ki67 expression is pretty high, and in the STS positive control, you can see much less Ki67 staining, but a significant higher apoptosis or cell death staining in cleaved caspase 3. And so here is the workflow that we trying to use to assess the treatment responses. First, looking at the MTS viability assay, and together with the histology and also IHC staining, then we decide or think whether it is responding or non-responding. And these slices can also be subject to further analysis like RNA-seq, looking at the transcriptional change, and also multiplex IHC, looking for the immune profile, immune component in the slice. And then we can eventually correlate this change with whether the slices are responding or not to a particular drug. And to summarize what we've learned so far, there are pros and cons for this system. The pros are that it is very clinically relevant because it's a patient-derived model, and it can retain tumor architecture and microenvironment. And we can maintain the viability for like 2 to 3 weeks, which allow screening of the drug. And also the fact that the slide culture system allows us to test multiple drugs simultaneously by single or combo drug treatment, and also allow further downstream testing. The cons are actually also related to the pros, because we see quite a bit of variation between the sizes based on the different tumor composition in the size. So in the second cause I want to highlight is that we observe that chemo-pretreated tumors do not maintain viability as the naive one. So the viability gone down pretty quickly within the first week. So we try to exclude these in our testing. In order to overcome some of the variation issue, we are working on organoid system to overcome the variation issue. And moving forward, we want to apply this system into two major focus. First is to develop into rapid drug testing system. Second is to try to use as a tool to validate our mechanistic finding. And we also want to invite more collaboration with other centers or group that want to develop this system, like especially we have so little case. And we also want to collaborate with anyone if they want to test some new drugs. We are happy to collaborate. And so thanks for the Cholangio Foundation for the award. And yeah.
Thank you very much.